Hello, everyone. Happy New Year. Welcome to the first National NBSTRN National Newborn Screening Pilot Webinar of 2022. We're going to get started. Seems like we got most people to join already. If you haven't done so, please mute yourself upon joining the webinar. This is a open forum discussion. So if you have any comments or any questions, feel free to unmute yourself and ask those, or you can ask them in the chat box. But if you don't mind, please mute yourself when you join the conference call. I also want to let everybody know that this webinar will be recorded and is also posted to the NBSTRN YouTube page. So if you colleagues happen to miss it, you can always share it with them at a later date. So we're going to still go through our standing agenda the same as we did in previous calls. So welcome everyone. If anybody is new to these calls and they would like to introduce themselves, please unmute yourself at this time and just let us know your name and the company organization you're representing would be great. Richard, you go ahead. Can you unmute yourself?
I'm unmuted.
All right.
Thanks for inviting me. I'm Richard Perrott. I'm a neonatologist at Brigham and Women's Hospital in Boston. And this is the first meeting I'm joining, and I'm representing a DMD newborn screening pilot that's taking place at Brigham and Women's Hospital.
Well, welcome to the call, Dr. Prod, and we actually have a little, I'm going to give away my slides, but we have an announcement about a podcast that you recently did in our slides as well. So thank you and welcome to the call.
Thanks.
Anyone else would like to introduce themselves? Okay, we'll go ahead and move on. Before we start with the pilot study states, I just wanted to announce that in a couple months, the ACMG will be hosting its annual clinical genetics meeting from March 22nd to March 26th. It is in person in Nashville, Tennessee, but it will also be virtually as well. So we have both options. NBSTRN will have a booth, be hosting a booth 736. So if you're at the meeting, please come and visit us. And we will also be hosting a scientific session on March 25th, the last day of the conference. Called Exploring the Role of Medical Genetics and Genomics in Advancing Newborn Screening Research. And we have three speakers that will... I'm sorry, Tara, did you have something to say? Did you have a question?
Nope, my apologies. My computer went off mute. Thank you.
Oh, that's okay, that's all right. We have three speakers that are going to be presenting during the session: Dr. Cynthia Powell, Dr. Deborah Friedenberg, and Dr. Beth Pletcher. Did someone else have something to say? I was going to see if any of our team members on the call had anything, other comments that they wanted to make. I think Dr. is on the call.
Did your thing freeze up like a minute ago or a couple minutes ago or while you were on?
No.
If you haven't already done so and you don't have a comment, please mute yourself. I'll try to mute the ones that you forget. Dr. Chang, did you have any comments that you wanted to add? I'm not sure if Dr. Brower is on the call as well, but she will be presenting a presentation called Secondary Data Analysis. Secondary Data Analysis, Opportunities to Advance Genetic Disease Understanding in Newborn Screening Research. And that will also be on the 25th day of the conference. I believe the the first session is on the 26th, or March 26th, and someone correct me if I'm wrong, if that session is, yep, it says it on my slide, the 26th. And then there will also be a poster session about a new project that NBSUN is conducting that's funded by HRSA. Well, it's a project with ACMG called Advancing Genetic Disease Understanding, Improving Health Outcomes. And it's a long-term follow-up project, which is called LTF Cares and LTF Check Initiative.
What did I do?
So now we're gonna, if there's a comments or any questions about the report, the conference, we hope that you all can join us. I'm gonna move on to the reports from the pilot sites. I'm gonna start with New York. We have the information in red that was reported in December, and any updates that you wanna give us for February would be great. So is any of the New York folks, I think I saw several on the call earlier. I see Michelle is unmuted.
Hi, everybody. Yeah, it's my turn this month. we rotate the report. So this is data up through, for all samples that were released from our program up through January 31st of 2022. For, which one I got here first? Oh, SMA, I'll go in your order. SMA, we've screened about, let's make sure I get it right, about 716,000 infants. We've referred 39, 3 since our December call. So the frequency has been fairly steady. I don't know if you have it on the other side there. No, so the frequency is around 1 in 18,400 for the incidence. Okay, you have the numbers on the copies there. Okay, so we had 1 baby still with 1 copy. We the 2 copies, we have 22 babies. So all 3 in the update are 2 copy. And then the other numbers are the same. So 11 with 3 copies, 3 with 4 copies, and 2 with 5 copies. Basically the same as you have over there in red.
Okay.
All right. ALD you have next, that's up here. Okay. We screened about 1.9 million infants still. There's 138 referrals, and that's 2 new ones since the December call. We have 69 boys and 68 girls. Update there. 50 of them had a variant in the ABCD1 gene. Uh, 42 have a diagnosis of ALD. And 15 Zellwegers. Or 15, I think, just with Zellwegers, yeah. Pompeii, we've screened about 1.65 million infants. We have 1 new referral, so we have 195 total referrals. And then that one was a possible late onset, so that's 106 possible late onsets. Yeah. Carriers are the same. The infantile is the same. Yep. For MPS 1, we've screened about 760,400. We have 37 referrals, which is 2 more since December's call. And we still have no confirmed severe, early onset MPS 1 babies. And we have one uptick, 6 total with a possible late onset, 2 variants identified. And the last one's GAMT. Same number, about 716,000 infants screened. We have no new referrals, so everything is the same from December.
Any questions for Michelle? Thank you, Michelle. Let me know if you guys have any questions, but we'll move on to the North Carolina group. I think Kate will be reporting out for that group today.
Yep. Hi, Jennifer. This is Kate. So for DMD, we've had 8,969 babies screened. We have 62 screen positives based on the one newborn specimen that we do, and 51 consented to genomic testing. Of those, we still have that one DMD case and a few carriers of other variants. So we're using 86 gene panel for this. And then we had 45 consents to latent CK testing. So that's an additional blood draw at 1 month. And 37 of those were normal range and 1 was slightly elevated, and the rest either were unable to get a specimen or they pending.
Can you say that one more? 37 were normal, then there was a few that were pending, and then
There is 1 slightly elevated, and the rest were either we were not able to get a specimen or they're pending.
and then the when you said there was carriers, I'm guessing these are variants of other genes. Is that correct?
Yes, we don't have any DMD carriers.
Got it.
And we have no numbers for MPS II. We're in the prep period. So we have some aggressive deadlines that we hope to meet. We really have, done most of the administrative things, IT security and things like that. We have acquired some shiny new equipment for the lab. And a lot of those things are new for us. As you know, we have a unique situation where we're testing at RTI and getting the specimens from the state laboratory. So we're working through some of the data and specimen logistics, and in the process of validating the assay. So and setting up follow-up on all those things. So no numbers yet.
It looks like you're making progress. So you guys, from last call, you have instrumentation, and you guys are starting the validation, correct?
We have one of the 2 Q sites there, so we're still waiting for the other one that should be arriving soon. And so, yeah, we should be ready to do the validation sometime soon, hopefully.
And I'm not sure if you have this information, Kate, or someone else on the call, that as far as the SMA screening being conducted in the North Carolina state lab.
Yeah, I do not have numbers for the state lab for SMA.
I saw Dr. Powell was on. I don't know if Kristen's on, and if they could potentially give an update. If not, that's fine too.
Yeah, I don't have that either.
Okay, thank you, Dr. Powell. Any questions for the North Carolina team about their the DMD screening? I know there was a presentation that was given, I think at the last year sometime, that gave an overview of the DMD screening process. So if you get a chance, check that out on YouTube. But any questions about that, as well as they're starting a new pilot for MPS 2?
Can you remind us how long your DMD pilot will go on for?
That's TBD. That depends on the funding that we have and that we might get. So we currently do not have a end date, a firm end date. So we're hoping to, we thought we would finish sometime this year, but we're hoping we'll be able to extend that.
Thank you.
Funding, I'm guessing, is not based on the number of babies or anything like that, Kate.
We do not have a target number or anything
okay.
like that. No.
Kate, for MPS 2, are you doing that like early check? Is that going to be part of your early check, or is that a totally separate consented study?
That is a separate study, and we're seeking waiver of consent.
Okay, thanks.
Okay, thank you. Any other questions for the North Carolina team? If not, we'll move on to Georgia. And I'm not sure who's on from that group. Is Angela on?
I'm here.
Great.
Hey.
Happy New Year.
Happy New Year to you guys. I apologize. I did not prep these numbers ahead of time. But I can tell you a little bit. SMA, we definitely have had one diagnosis since our last conversation, but that makes about 7 since we ended the pilot. It's kind of hard to count the way the numbers are listed in front of me. But it's going well, and we don't really get false positives. We certainly do get, inconclusive results, and it's usually also, it flags SCID, and then but the internal control is abnormal also, so those we just repeat screens on them. But that's about it for SMA. Let's see. Okay, MPS 1 is easy because that's the one of the ones we're sending out a second tier from the lab. The second tier test for GAGs is going up to Mayo, right from the dried blood spot. It's going really well. I know they've been sending spots up, but we have not had any abnormals from that. So hip hip hooray. And then let's see, Pompeii. We're just doing enzyme. Let me look over here. Am I looking at the right one? Yes. Okay, Pompeii. We have had 1, 2, 3. Oh, here, 9 abnormal results. Screens, sorry. 2 of those kids turned out to be normal. We have 1 kiddo who is diagnosed with a later onset Pompeii disease. And then the others are pending. That's 1, 2, 3, 4, 5, 6 are pending. So we will figure that out. Let's see. ALD is what I'm looking at next here. Sorry, I have multiple screens open here. So for ALD, since we restarted in 2020, we've had 32 kids who were abnormal on screen. And then we've had, let's see, 18 minus, hold on one second. 1, 2, 3, 4, 5 of those 18 were diagnosed with Zellweger syndrome, and the remainder were XALD.
Okay.
And I'll wait for you to catch up. Okay, and then the Krabbe pilot is very similar to MPS 1. Also, a second tier test for cycosine going off the dried blood spot to Mayo. And so again, we've had a good handful of spots go up and no positive results from that. So we no real referrals from that. So far so good. And then PUCD pilot, I think we already told you, wrapped up already. We've just submitted our report to the NIH, and we are working on writing a manuscript from that so that it'll be more widely available. And homocystinuria, we're still gearing up. And then I could go on and on and on from there, but that's not so interesting.
No worries at all. That's great. Any questions for Angela about her results? I think you did say that the Crabbe, Pompeii, and MPS were kind of started at the same time, correct? I think you said that on a previous.
Correct. I think it was September, if I'm remembering correctly. September 2021.
Okay. September 2021. 20.
Yeah. Yeah.
Someone have a question?
Yeah, Cindy Powell from North Carolina. Angela, I was just wondering, for your MPS 1 protocol, you're sending the dried blood spot gags to Mayo. Is that, if those are negative, then would that be considered a negative screen? Or if there's still like a really low enzyme level, are you still referring those? Even
no,
with
there's no like ultra low enzyme that we have talked about anyway. Nothing that I've heard about.
That. Okay, so you're going off the dried blood spike gags then.
Yeah, we just got too many pseudo deficiencies in this state.
Yeah. Okay, thanks.
Yeah. Jalili, let me look at that for you. No, I think it was 18 that were true abnormals. Hold on one second. Oh, I closed that tab. Give me one quick second and I will tell you. I'm pretty sure it was 18 and then 5 were Zellweger, but
And while you're looking it up, I'll just read you Lily's question. It says, XAOD in Georgia confirming you have 27 true cases since 2020. So Angela,
15, sorry. Did I type that in right? This is what happens when I rush things. 2020, 2022. Yeah. So, yeah, 15 positive or 15 diagnosed cases. That can't be right. Hold on. Let me look at what I told you.
You said 5.
I told you 5. Hold on a second. That's not right.
Okay. No worries. No worries.
This is why I should prep ahead of time, right?
It's totally okay. We're just happy to get the numbers.
No, I think this is right. Yeah, no, we started, it's not 5, it's 3 Zellweger and then 12 ALD.
Okay, so you might either have some pending cases or normal screen.
Yeah, everybody else would have been closed as, let me make sure this is right. Give me one quick minute and just clear the form because sometimes I have things that are hiding from me. ALD, Zellweger, come on, work for me. And we started in 2020. Search. Yeah, that's right. That diagnosed case is right. 3 Zellweger, 12 ALD. And then just double check the referred cases. Yeah, 32 that were referred as abnormal. We have a mix of things, some that were resolved as normal, a small handful that are still pending. Like we have 1, 2, 3, 4 5 kids that are at least still pending. 1 kid was lost and a few that were normal. So we get a few false positives on that, but not a ton.
Okay. And I'm assuming right now, most of the 12 cases that are confirmed with the diagnosis, you're just monitoring right now?
Yeah, what we
That's
do is we do an
challenging.
immediate referral over to endocrinology, just to make sure that we catch them before there are any problems and get them in that system. And then we also make a referral to pediatric neurology at that point. Genetics doesn't follow up with them too much at that point. I say we because I sit in genetics, sorry y'all. But so we do follow up if we need to, more for genetic counseling and things like that. But mostly we hand the care over to endo and neurology, and then neurology will refer as needed for bone marrow transplant.
Sounds good.
And Bill, in case you have anything else to add on that, but I think that's generally how that works for the ALD kiddos.
No, that's pretty good. I would just add that homocystinuria startup is waiting, I think at this point, for a new lab director in the state, because Trisha's moving to Mayo. She'll still work with us on the projects, but we've got to have a new permanent director to start that thing up.
Well, I know Patricia will be missed in your program.
Yeah, if anybody
Absolutely.
wants to move to a nice place that's doing pilot studies and has more temperate weather, apply now.
Great team to work with.
We're really great people.
All the pilot sites are great teams to work with. Great. Any other questions for Angela or Dr. Wilcox? Just let us know. You said temper weather, right? Kim Piper was saying the weather is probably better than most places right now.
It is 60 degrees right now outside, just so y'all know.
Yeah, rare snow that doesn't stay on the ground too long, and summers are kind of warm, but it's not bad at all for the U.S. So
yeah, everybody that's having horrible weather right now, stay warm, bundled up. I know it's been difficult. It's been a very interesting winter as far as January. So, and then I think Jalili just posted in there the job announcement if anybody's interested in the Georgia position.
Yeah, are you gonna email me too, or Trisha, and find out about it, what it's like down here?
So with that said, if there's any other states that want to report off what's going on, if they're part of new pilot studies, that would be great. If they are starting to screen for one of those, I guess, more recently added conditions, SMA, MPS1, Pompe, XALD. Now we have the 2 nominations for GAMT and MPS2. If you guys have started screening for that, or if you have any questions that you would like to discuss on the call, please chime in, unmute yourself, and let us know. Or you can enter something into the chat box as well. Oh yeah, Dr. Prad, please unmute yourself.
Well,
If you want to
officially,
give us information about the DMD pilot you're conducting, that would be great too.
I can. I didn't know exactly what to expect here, so I didn't come with the latest numbers, but I can give you some numbers. And we're actually not a state, so we are a hospital. So we're a little bit of an anomaly here. The program is run entirely from a single birth hospital. It's the largest delivery service in the state of Massachusetts. We have about 7,000 deliveries a year. We are offering this to families free of charge. And we started mid-July of 2021. We've screened, as of last week, we had screened 3,264 babies. We have an algorithm of one dried blood spot, CK, whole exome sequencing, looking only at the DMD gene. And we had 82 babies who had elevated CKs whose samples were referred to genomic screening. We've had no detections to date. We have been getting repeat CK on babies after the whole exome sequencing. This is our follow-up. Neurologist want us to do this. I think we're probably not going to continue this very long. But just to, we're only consented to look at DMD. We do have potentially information on other genes that might be causative for elevated CK. So we've been doing sort of CK, next generation sequencing CK. And if the second CK is elevated, then we'll go back and get consent to look at the other information that we may have. But none of our repeat CKs have been elevated. So and one of them was very, very, 1 point over the mark. So that's where we are right now. I'm happy to, share the information regularly with the group. And I'm also hoping, I'm in the process of trying to get an agreement signed, a disclosure agreement signed with BPMD that we would be able to help and contribute to their efforts towards a RESP submission to add our data to the mix with New York and North Carolina. Our algorithm's a little different from everyone else's, so hopefully we can contribute. And we don't have an end date at the moment. We are funded and we're continuing as long as we can continue to squeeze some funding dollars into the program.
Thanks, Dr. Parad. That's so exciting about your effort. Can you tell us a little bit about your recruitment process?
Sure. We have made the decision in discussion with our clinical staff that this is not it's a pilot, but it's not a research project. The literature on CK screening for DMD is almost as old as I am. So I think we know we can detect babies at risk for DMD, but I think it's because of all the new therapies that have become available that would be appropriate for some of those babies, that this was felt to be really offered as a clinical option for parents. So at the as you probably most of you know, Massachusetts has a pilot program which currently includes I think 9 disorders, and parents are asked after from their general pediatrician in the postpartum floors. They are asked whether they want to participate in the state pilot when the newborn screen is drawn on their baby. And we have sort of piggybacked onto that. After that question is asked, they're asked if they are interested in having this other supplemental test for DMD. And if they are interested, then they do sign a consent form. It's a consent as in having a genetic test done, which explains the test and the risks and benefits of performing the test. And then if they say yes, and a separate filter paper card is collected at the time that the newborn screen Guthrie card, the state mandated Guthrie card is collected, so that the baby doesn't need to have an extra blood collection. And then we're totally an independent process. So we send the samples off to be sequenced. We did publish a paper in IJNS few months ago describing our process. We're using the Perkin-Elmer CKMM assay. So we send to them, and if they're above our cutoff, then they reflex to actually whole exome. So that's how we offer to parents, and about 80% of our parents say, yeah, I'd like to do that.
Thank you. And we have several questions in the chat, and I think you answered the last one from Dr. Kajana, which is, are you using CK or CKMM? So it sounds like you're using the Perkin-Elmer CKMM assay, correct?
Correct.
And
other questions there? I'm looking, so I see
yeah,
now.
there was one about how many of the approximately 3,000 babies that were screened are sent for sequencing. Do you happen to have those numbers?
Yeah, so it's been 82 out of 3264. You'll have to do your own math.
Okay.
And I also see from Dr. Powell, I see, will we be enrolling them in a clinical trial? So we, as part of setting up this program, we were fairly thoughtful about, because of what I have found to be a lack of clear guidance in the literature or evidence base about exactly what therapy should be initiated should a newborn be detected with DMD. We developed a work group, including, and so we're connected to Boston Children's Hospital, where there is a DMD and neuromuscular clinic. So they have pediatric neurologists who are specialists in neuromuscular disorders. We've hooked up with them and representatives from genetics, and so we meet regularly. We've been we actually are hoping to publish a paper on this fairly soon, or submit the manuscript pretty soon, on kind of reviewing what's known out there about what you would do for these babies. So at the moment, we would refer them directly to our follow-up clinic, and we have developed a follow-up protocol based on what we know so far. And it would be up to the neurologists as to whether, well, first, based on the testing, there might not be an appropriate new FDA approved drug yet. Steroids, most people are not starting below 18 months, or some people will start them a little bit earlier. But there's a lot of follow-up and certainly neurodevelopmental follow-up that may be appropriate to get them hooked up with right away. And I think that, Cynthia, I think that was your question.
Yes, thank you. Guidelines would be great, I think, because we're sort of running into similar things here. We don't, with the follow-up for the North Carolina babies. So, I mean, it's fairly small percentage that would qualify for an FDA approved treatment, I think. So
I think it's about a third of them. So it's definitely the minority. Yeah, we've actually thought about maybe trying to do sort of a Delphi approach to this, of the pediatric neurologist who take care of DMD children, to see if we could use that for some evidence on which to base our proposed follow-up protocol. It's clear that some people, some neurologists want to start these FDA approved drugs if they match right away, and there are no FDA restrictions on age for those new drugs. And there are a number of other drugs in the pipeline that hopefully will be filling the gap and getting the percent treatable up to a higher percent.
right. I mean, certainly for RUSP consideration, the benefit of early diagnosis in terms of what treatment is given is going to be really critical. So
yeah,
it's great that you've got this one going.
we're trying. It's not an easy answer.
I know. Thank you, though. Glad you could join us.
Thank you.
So there's one question I have, and then there's another question in the chat, which is a good one that Ryan asked. So you said CKMM is done on the 1st, and I'm guessing this is on dried blood spots. After, if the DMD, if you don't find any variants, do you do another CKMM, or are you doing a urine total CK at that point?
So we're doing CKMM on the first dried blood spot. The DNA is extracted from that same first dried blood spot, so we don't call anybody back. And before we report out the results, we wait for the sequencing results to return, and then we contact the family afterwards about having the repeat, and the pediatrician about repeating the CKMM at some point in time. There aren't normal values for the Perkin-Elmer CK assay outside of the newborn period or the first 20 days, then it's 60 days. So really that second level, we ask them to try to repeat it on the baby's 2-month well child visit, and we send out a kit to the pediatrician's office or the family for them to bring in, so they don't have to pay for that. We've interestingly found that the hang up there is that lots of pediatricians' offices do not draw blood or do heel sticks anymore.
Yeah.
And so this has gotten more complicated than we anticipated. And really it's being the neurologists were being, extra careful, because we were getting the whole exome and we had that potentially additional information. We are considering, backing off and doing just DMD sequencing, and then we will have answered our newborn screening question, and we wouldn't do the follow-up CK. So I think our assay is probably going to change, our algorithm is probably going to change to just getting one initial sample and doing a CK followed by a DMD sequencing, and not any follow-up. But that's a decision in process right now.
Yeah, I remember when I was working with the Carolina group, Dr. Powell and I, we had discussions about the SMA pilot and the same problems where a lot of pediatricians couldn't do a blood draw in office, and how was that going to happen? So Ryan had a question also, just wanting to expand on the discussion using whole exome versus whole genome versus a target panel for follow-up. And you kind of hit on that already about I guess there's a particular clinician that was very interested in opening that panel up, but you guys are going to probably move away from that process.
We used a whole exome mainly because the lab that was providing us an incredibly good price for doing the sequencing, that was what they do in large volumes. They do whole exomes on dried blood spots. So we couldn't really modify that if we wanted to do whole genomes, which would have been, as a little sledgehammer opening a walnut. I think that would have been more expensive, and we didn't have the funds to do that. If we would have been able to choose just doing DMD, we would have done that. It just wasn't an option from that laboratory. So we sort of have an in silico filter where we are only given information about DMD, but the data are there for the other neuromuscular genes, and we have not had any reason to go back and look at anything else to date. But our numbers are, pretty small so far.
And then there's another question by Dr. Vogt about, do you offer pilot testing parents of female newborns? Is it to everybody?
Yeah, good question. And we just thought it was too complicated to try to segregate sexes. So we offer it to everyone, and the consent form says this is most likely to affect males, but rarely can be an issue for females. So parents are aware of that up front, but we offer it to everybody.
Thanks for that information. Is there any other questions that anybody has? This has been a very interesting discussion about that, and we're looking forward to hearing some updates, I guess, in our future calls. That would be great.
Happy to do that.
I know that Arkansas also commented, I'm scrolling back through they has started screening for SMA and XLD at the beginning of the year. SMA, they identified 2 over the summer during the validation, and 1 positive in January. For XALD, they've confirmed 1 that was for genetic testing. So if anybody is I'll copy this note, but if anybody is from the Arkansas, Fran, if you want to unmute yourself and share more about your screening experience, that would be great.
Sure, and it's Arizona, not Arkansas.
Oh, I'm so sorry. I'm so sorry. You didn't say Arizona.
No worries. I think what I put in the chat box says it all. We've been screening now officially for a month, and so we've had 1 positive since officially starting testing in January. Baby had 3 SMN2 copy numbers. And then just the 1 with the presumptive positive for XALD, and we're waiting on the gene testing to come back.
And I'm assuming the gene testing is happening with a clinical follow-up. It's not
correct. We don't do the ABCD1, so we're waiting for that to come back from the follow-up geneticist. And then during our validation study over the summer, we had the 2 positive for SMA.
And were those also just referred out, or did you have a confirmation from another newborn?
For the SMA, those were referred out for the SMN2 copy numbers. I don't have offhand the copy numbers, but it was early onset, and they both received gene therapy within about 2 weeks. So
that's great. Sounds like everything's going well in the program.
We're doing our best.
Yes. If Dr. Schroth is on, I'm not sure if she's on or not, but it seems like a lot of most of the programs have they started screening for SMA? Are we up higher now than before for last year? I'm not sure if she's on the call today. If anybody has any information on that, it'd be interesting to know, to see how many states are actually screening from SMA now. And Ryan, if you want to unmute yourself and ask your question, that would be great. I think you said, curious, what's the cost of cost sheet? A lot of discussions about trajectories of different technologies. I'm guessing his question's about NGS. Dr. Vogt asked about many newborn screenings finding the 3. Dr. Vogt, did you want to unmute yourself and ask your question? Finding of 3 positive cases. Oh, Ryan's on the plane.
This is Fran in Arizona. I'm assuming he means the 3 positive cases we have identified in Arizona so far. I believe that our validation study was about 7,000 samples where we found the 2 positive cases. And I'm not sure how many samples we ran in January before that that baby flagged positive. But it was at about the middle of the month. So I would guesstimate there were only a few thousand samples run prior to flagging positive our first one of the year.
This is interesting. Seems like your incidence rate seems a little bit more frequent, if that's the case, than some states.
Jen, did I unmute? I can't find these.
Yeah,
buttons.
we can hear you, Dr. Vogt.
Okay, well, I hope I can figure out how to mute myself again. So that could you all at NBSTRN, it might be a good time sometime this year to collate all this data, because some people are saying, oh well, that I actually, there was a paper that used in the title used the phrase low incidence, and I think lower would have been better than low, because by newborn screening standards, 1 in 20 or 30,000 isn't really low, I guess, but it's higher than the 1 in 10,000, which was the sort of fictitious number that people were working with. But in some cases, it looks like it might be higher. I mean, if we wanted to settle on 20,000 as some sort of benchmark, it might be higher than that, but you need millions of babies to get that that confidence interval to make any sense. So I'd like to lobby for a concatenation of data from all the programs that have been screening so far, and let's get a reasonable estimate of the what I call the birth prevalence of SMA in the United States.
I think that's
a great. Oh, go ahead. Jen.
No, go ahead, Amy. Go ahead.
I think that's a great suggestion and something we've been thinking about, as we get all this great data on these calls. But again, it's just a snapshot, and it's only the programs that are on the calls. We know APHL New Steps has case definitions for the RUSC conditions, and they collect data from some state newborn screening programs. But I think, Bob, your point is well taken. And what we can do is connect with Jalili and his team and figure out a plan going forward where we can represent some of this data, and knowing that, it's emerging data, and that, again, it's snapshots of time. But, we've been really struck by the data as well. And we learned so much by these helpful calls and the reports from the state programs.
Yeah, well, that would be great, Amy. If there's any way that I can help with that, or we can help with that, please let me know, because at this point we must have, I guess, millions of newborns that have been screened for SMA, I think, maybe, or 1 million or something like that. And it would be possible by concatenating to get a reasonable estimate, I think, that can be improved over time.
Well, this is Stan from California. We're putting together a paper on SMA. So I think we're approaching 1 million.
Yeah, well,
individuals.
California,
yeah,
make these estimates.
Yeah, and then we could, yeah.
Do you want to preview what the paper might say?
Not today. I can't say anything really. I know. Well, it's not me. So let's just keep it at that.
All right. Well, I was trying to be silly in a way, but it is.
I know.
It probably the original 1 in 10,000 is probably too high, but it's probably overall more than 1 in 30,000. I'm going to guess, but who knows? So. The sooner we get some kind of reasonable estimate there with confidence value intervals that make sense, that'll be very, I think, beneficial.
Thanks, Bob.
Yeah, thanks, Bob, for that. And we'll note it sounds like there's multiple partners involved in that discussion. So we'll continue that discussion. But since we're coming to the end of the hour, I just wanted to highlight a few more slides for everyone. And if Dr. Chen is still on the line, feel free to jump in, or Amy, Dr. Brower, you can feel free to jump in as well. Please register. If you could go ahead and register on the NBSN website. We're sending out newsletters about upcoming events or things that are happening in the community. So please effort. And I think that Is that letter sent out monthly, Dr. Chen or Amy?
Yep, it's a monthly newsletter, and if you have any events locally or regionally you'd like us to highlight, please feel free to message us.
And then the last slide is January, our episode 8 of our podcast, Newborn Screening Spotlight, was released. So please feel free to also log onto our website and listen to the podcast. Dr. Prad is one of the speakers on the podcast. He's talking about, he was one of the principal investigators for BabySeq, which was a 5-year genomic study. And then he also leads the DMD newborn screening program that he just spoke about. So feel free to please, you can get this podcast wherever you listen to your podcast. So Apple, Google, Spotify, Amazon, iHeart. And then this month, I think they're releasing a podcast where Dr. Wilcox will be speaking about his pilot study experience, if that's correct. Is that correct, Amy?
Yep, we've got a lot of heavy hitters coming up, so we're excited about that. Thank you.
Yeah, the podcasts are really interesting. We've had past one where Dr. Howell has spoken about the history of newborn screening. We've had an SMA parent that's given some information. I think, so we've, Dr. Schroeder has presented as well. So we've had some great speakers. So please join in and listen to the podcast. And then lastly, we have these calls every Thursday at 2 p.m. Eastern Standard Time. So the next call would be March 3rd, 2022. So thank you all for joining again, and I hope you have a good rest of your day.